Anunnaki, not these other brothers. I ain't with that black community shit.
African genetic continuity in Egypt from the Stone Age to the Dynastic period. The case for the African EM215/M35 and its many descendants. The first question that should arise is, when did the Stone Age end? Even though I could talk about 300,000 years ago in Egypt, I don't have to. According to S. Harmon et al., 3.3 million year old stone tools, the Stone Age is a prehistoric period when weapons and tools were made of stone or of materials such as bone, wood, or horn. This period lasted millions of years and came to an end around 10,000 or 12,000 years ago. This debate is about genetic continuity from prior to this window through the pre-dynastic and dynastic periods of ancient Egypt. In order to be a Stone Age people in these areas, there should be Stone Age genetics and technology in the areas in question. Is there? Yes. There is, in the form of Y-DNA haplogroup M215/M35, mtDNA haplogroup L3, and their many descended subclades, among other archaeological evidences. Early Egyptian weapons. In the early dynastic period in Egypt, circa 3150 to 2613 BCE, military weaponry was comprised of maces, daggers, and spears. The spears had been developed by hunters during the pre-dynastic period and changed very little except like daggers. The tips changed from flint to copper. Even so, the majority of spear and arrowheads from the Old Kingdom of Egypt seem to have been largely flint. An Egyptian soldier would have carried a spear and a dagger and a shield, probably made of animal hide or woven papyrus. These weapons were supplemented during the Old Kingdom by archers who used a simple single arch bow with reed arrows and flints or copper tips. The fact that pre-dynastic Egyptians were using flint arrowheads is evidence of them being a Stone Age people, as flint is a type of stone. The evidence of the evolution of stone arrowheads and spear points in these areas dates back to the Nubian lavello technology of early humans over 250,000 years ago. Its evolution in these areas until early dynastic periods is evident of cultural continuity. It's clear that pre-dynastic technology had Stone Age roots inside of Africa. Stone Age genetics. All it takes is a simple Google search, folks. Y-DNA haplogroup EM35, a branch of the broader EM215 lineage, is estimated to have originated in the Horn of Africa around 23,000 years ago, 23,000 BCE, during the Stone Age, with its most recent common ancestor likely born between 26,042 and 19,538 BCE. Phylogeography of haplogroup E. Phylogenetic information and the geographic distribution of haplogroup E clades, we performed a discrete phylogeographic analysis, U et al. 2010, to associate each node of the tree to one or more of 6 broad geographic regions with an emphasis on the African continent. This analysis suggests a sub-Saharan placement of the most common recent ancestor of the haplogroup E chromosomes here analyzed. Within this clade, the posterior probability strongly favors an East African placement for the origin of EM215 diversity, as previously suggested by Seminole et al. 2004 and by Gebremescu and Ibrahim 2014, whereas a Northern African location is favored for the node defining the M78 subclade, supporting the previous hypothesis. Despite we assign most previous deep EM35 Eastern African clades to haplogroup EV1515. Our phylogeographic analysis slightly favors an Eastern African origin for M35. We found a new clade defined by the V1515 mutation, which originated and differentiated in Eastern Africa and expanded southward in recent times as a single terminal clade, EM293. We propose that the migration marked by the EM293 haplogroup could be the final step in a north to south range expansion listed as linked to different branches of EV1515, which initially involved people from Eritrea and possibly northern Sudan not sampled here. This migratory route is concordant in time and space with archaeological evidence for early domestication of African cattle in northeastern Africa about 10,000 known years ago. Southward climate-driven movements of herders into southern Ethiopia highlands and Turkana Basin, northern Kenya, about 4,000 years ago, and a subsequent sub-equatorial pastoralist expansion towards southern Kenya, Tanzania, and southern Africa not before 3,000 years ago. Some controversy persists in the archaeological community regarding whether cattle domestication was developed in North Africa or imported from the Middle East. However, recent DNA analysis of extant indigenous African bovine, taurine, and zebu cattle, Harnott et al. 2002, supports a model in which the earliest emergence of pastoralism involves taurine cattle took place in northeastern Africa and subsequently spread westward and southward. Harnott et al. 2002. A recent analysis of NRY variation in 13 eastern and southern African population samples suggests that the spread of pastoralism from eastern Africa to southern Africa was accompanied by a migration of pastoral peoples as well as pastoral technology, as evidenced by the distribution of NSR haplogroup 293 and the subclade E3B1F M293, Hen et al. 2008. Furthermore, the most likely source for this migration based on the samples included Hen et al. 2008 would have been the Southern Nilotic speaking Datog, because of the haplogroup frequency and diversity of M293 is highest in the Datog, around 2,000 years ago, Hen et al. 2008. Earliest evidence for Egyptian cattle is from the Fayum region, dating back to the 8th millennium BC. In the past, the types was given its own species name, Bos A. aegypticus, but is now regarded as part of the same scientific taxon as other domestic cattle, either Bos taurus or Bos primigenius, subspecies taurus. It is uncertain as to where Egyptian cattle originated, as some claim that they were acquired from the Levant, Mesopotamia, while others claim that it was domesticated independently from a North African subspecies of the aurochs, Bos primigenius mauritanicus. There's evidence for both sides, as cattle has been domesticated in the Levant by the 8th millennium BC, but excavations of early Holocene Western Sahara shows that indigenous cattle existed previous to the 8th millennium. Spread of pastoralism. Archaeological data suggests that the emergence of animal husbandry in northeastern Africa took place as early as 11,000 years ago. Mendorf and Skilled, 1998. Archaeological studies in Apta-Playa in Egypt's western desert reveal a spectrum of artifacts consistent with pastoralism and adaptation to the desert environment, including particular pottery styles, Khartoum tradition, evidence of well technology, and cattle burials. McDonald, 1998, Windorf, and Skilled, 1998. By around 8,000 years ago, evidence is presented of imported in the Middle East sheep or goat remains in northeastern Africa. From McDonald, 1998. Although geneticists have extensively debated the mode which agriculture diffused from the Near East to Europe, they have not directly examined similar agro-pastoral diffusions in Africa. It is unclear, for example, whether early instances of sheep, cows, pottery, and other traits of the pastoralist package were transmitted to southern Africa by endemic or cultural diffusion. Here we report a newly discovered Y chromosome-specific polymorphism that defines haplogroup E3B1F-M293. This polymorphism reveals that the monophyletic relationship of the majority of the haplotypes of a previous paraphyletic clade E3B1-M35 is widespread in Africa and southern Europe. To elucidate the history of the E3B1F haplogroup, we analyzed this haplogroup in 13 populations from southern and eastern Africa. The geographic distribution of the E3B1F haplogroup is associated with the microsatellite diversity estimates for populations. It is consistent with an expansion through Tanzania to southern Central Africa. The data suggests that dispersal was independent of the migration of Bantu-speaking peoples along a similar route. 11,000 years before present marks the end of the Paleolithic, Old Stone Age, and the beginning of the Mesolithic, Middle Stone Age, and the start of the Holocene epoch, a period of transition to the Neolithic, New Stone Age, and the earliest evidence of cattle herding or pastoralism in northeast Africa is at least 3,000 years prior to West Eurasian sheep and goats being imported into Africa. Early African pastoralism viewed from Dakhla Oasis, South Central Egypt, M.A. McDonald. The late prehistoric archaeological sequence of Dakhla Oasis, South Central Egypt, is examined for evidence on the origins and development of pastoralism in northeastern Africa under the dry but fluctuating environmental conditions of the early to mid-Holocene. 8,100 BP, relatively sedentary groups the Masara cultural unit have a broad-based subsistence but no sign of food production. Herding appears circa 7,000 BP at a time of increase and possibly less seasonal rainfall on large late Bashindi A sites with stone-built structures and still diversified food economy. With the drying trend after 6,500 BP, mobile Bashindi B cattle and goat herders continue to aggregate in the oasis for a millennium, still utilizing a variety of resources. More settled Sheikh Mufta groups occupy the oasis lowlands until Old Kingdom times. Throughout the sequence, the early pastoralism from Dakhla seems more African than West Asian in character. Again, seems more African than West Asian in character. Not only does EM35 and its subclades have genetic continuity since the Stone Age, they have cultural continuity too. Pastoralism gave rise to the cattle culture of dynastic Egypt. Y chromosome haplogroup EM78, a descendant of EM35, a prominent paternal lineage in North and East Africa, likely originated in Northeast Africa during the Paleolithic era. Evidence suggesting migrations between Northeast and Eastern Africa, as well as trans-Mediterranean migrations to Europe and Florida West Eurasia. Detailed population data were obtained on the distribution of novel biallelic, finally dissect the human Y chromosome haplogroup EM78. 6,501 Y chromosomes sampled in 81 human populations worldwide. We found 517 EM78 chromosomes and assigned them to 10 subhaplogroups. 7 microsatellite loci were used to further evaluate subhaplogroup internal diversification. Graphic and quantitative analysis of haplogroup and microsatellite diversity is strongly suggestive of a northeastern African origin of EM78, with a corridor for bidirectional migrations between northeastern and eastern Africa, at least two episodes between 23,000.9 and 17,000.3 known years ago, and between 18,000 and 5,000.9 known years ago. Trans-Mediterranean migrations directly from northern Africa to Europe, mainly in the last 13,000 known years, and flow to northeastern Africa to western Asia about 20,000 years and 6.8 thousand years ago. A single clade within EM78, EV13, highlights a range expansion in the Bronze Age from southeastern Europe, which is also detected by haplogroup JAM12. Phylogeographically pattern of molecular radiation and coalescence estimates for both haplogroups are similar and reveal that the genetic landscape of this region is, to a large extent, the consequence of a recent population growth in situ rather than the result of a mere flow of Western Asian migrants in the early Neolithic. Our results not only provide a refinement of previous evolutionary hypotheses, but also well-defined time frames for past human movements, both in northern, eastern Africa and western Eurasia. So we see EM78 emerges during the late Stone Age in northeast Africa in the area around Egypt and Libya, and shows not one but two Stone Age migrations from northeast Africa into eastern Africa, both likely occurring prior to the end of the last glacial maximum. The study also determined that the genetic landscape of this region is, to a large extent, the consequence of a recent population growth in situ rather than the result of a mere gene flow from Western Asian migrants in the early Neolithic. In situ is a Latin phrase meaning in place or on site, referring to something remaining in its original location or context. The Stone Age presence of African e-markers is not only established in Egypt, but also in Western Asia. While haplogroup EM35 and its subclade EM78 are indeed both found and originated in Africa, they are primarily associated with North and East Africa, the Middle East, and Europe, not Sub-Saharan Africa, but they're both African nonetheless. Let's get to this 2017 Ebersir El-Malik study by Schunemann et al. You like to use. Here's what you're not telling the people about it. The affinity to the Middle East finds further support by the Y chromosome haplogroup of 3 individuals for which genome-wide data was obtained, 2 of which could be assigned to the Middle Eastern haplogroup J and 1 to haplogroup E1B1B1 common in North Africa. E1B1B1 is EM35. That's a 34,800-year-old Stone Age African haplogroup in your Middle Kingdom Roman period Fayum study, Joshua. Its direct descendant, EM78, emerged just west of the Fayum site over 20,000 years ago. Finally, we use 2 methods to estimate the fraction of sub-Saharan African ancestry in ancient and modern Egyptians. Both reveal modern Egyptians inherit 8% more ancestry from African ancestors than the 3 ancient Egyptians do, which is also consistent with the admixture results discussed above. Absolute estimates of African ancestry using these 2 methods in the 3 ancient individuals range from 6 to 15%, and in the modern samples from 14 to 21%, depending on method and choice of reference population. The study not saying that they had no sub-Saharan African ancestry, Joshua, you are. Finally, we analyzed several functionally relevant SNPs in sample JK2911, which had low contamination and relatively high coverage. This individual had a derived allele at the SLC24A5 locus, which contributes to light skin pigmentation and was shown to be at high frequency in Neolithic Anatolia, consistent with the ancestral affinity shown above. Other relevant SNPs carry the ancestral allele, including HERC2 and LCT, which suggests dark-colored eyes and lactose intolerance. Our analysis furthermore shows that the derived alleles for genes SLC24A5, known to be responsible for partially lighter skin pigmentation, were present in both JK2888 and JK2911. For further genes such as SLC45A2, LCT, and EDAR, we were unable to find derived alleles for both JK2888 and JK2911. For JK2134, there was no sufficient coverage after quality filtering at all the specific sites, which is why the analysis revealed no further clues. E1B1B1 refers to the human Y chromosome DNA haplogroup, also known as EM35, with an East African origin and associated with the spread of pastoralism from East Africa to Southern Africa, Joshua. It makes sense it was found in your study, seeing how its direct descendant, EM78, emerged just west of the Fayum Ebersir El-Malik site closer to the Libyan border in northern Egypt, long before the Neolithic expansion in these SLC24A5 mutation carrying Neolithic migrants, and is more evidence of African genetic continuity in Egypt from the Stone Age to the Middle Kingdom and Roman period your favorite study is based on. And then there's the more recent 2020 study of mummies at the Kurchatov Institute. In 2020, three mummies dating from the first millennium BCE from the Pushkin Museum of Arts collected were tested at the Kurchatov Institute of Moscow for their mitochondrial and Y chromosomal haplogroups. One of the mummies was found to belong to Y chromosomal haplogroup R1b1a1b or R1b-M269, which originated in Eastern Europe, and another two Y chromosome haplogroup E1b1, B1a1, B2, A4, B5a, which originated in North Africa. They also belong to mitochondrial haplogroups L3H1 and N5, common in Africans and Middle Easterners respectively. The third mummy was found to belong to mitochondrial haplogroup N, which is widely distributed across Eurasia as well as Eastern and Northeastern Africa. E1B1, B1A1, B2A4, B5A is also known as EM180, and the downstream snap EM180 may have originated in the humid South Central Sahara savanna grasslands of North Africa between 14,000 years before present and 10,000 years before present. Quit playing, Joshua. The expansion of mitochondrial haplogroup L3 within and outside of Africa. Genetic evidence from the extant populations have suggested that non-Africans descend primarily from a single successful later migration. Within the human mitochondrial DNA tree, haplogroup L3 encompasses not only many sub-Saharan Africans but also all ancient non-African lineages, and its age therefore provides an upper bound for the dispersal out of Africa. An analysis of 369 complete African L3 sequences places this maximum at 70,000 years ago, virtually ruling out a successful exit before 74,000 years ago, the date of the Toba volcanic super eruption in Sumatra. The similarity of the age of L3 to its two non-African daughter haplogroups M and N suggests that the same process was likely responsible for both the L3 expansion in Eastern Africa and the dispersal of a small group of modern humans out of Africa to settle the rest of the world. The timing of the expansion of L3 suggests a link to improved climatic conditions after 70,000 years ago in Eastern and Central Africa rather than to symbiotically mediate behavior, which evidently arose considerably earlier. The L3 mtDNA pool within Africa suggests a migration from Eastern Africa to Central Africa 60 to 35,000 years ago, and major migrations in the immediate post-glacial again linked to climate. L3H is a specific branch within L3. It's estimated to have originated around 58,000 years before present, with L3H emerging around 56,000 BCE. While L3H is associated with the migrations out of Africa, it's also found in various populations within Africa, suggesting the complex interplay of migrations and genetic diversity. That's a 58,000-year-old East African mitochondrial haplogroup found in the first millennium BC mummy, Joshua. That's 55,000 years worth of genetic continuity, and the best you got is its sister clade within L3, M1A1, and all of your Eurasian Y haplogroups are associated with the Neolithic ball. The 2018 study titled Carriers of Mitochondrial DNA Macro Haplogroup L3 Basal Lineage Migrates Back to Africa from Asia around 70,000 years ago had this to say about haplogroup E. There is general consensus in attributing an Eastern African origin to the initial expansion of the NRY haplogroup E in the continent. Curiously, ancestral E lineages have been detected in the Arabian Peninsula and the Levant. Regardless of its origin, haplogroup E shows lower frequencies in northeastern Africa and the eastern Sahel than in the west, The opposite pattern is observed in the frequencies of wide Eurasian haplogroups in the same areas, which suggests later, stronger Eurasian male gene flow throughout the northeast side. The study, titled Eurasian Back Migrations into Northeast Africa, was a complex and multifaceted process, gives us clues as to when. In the last decade, several genetic studies on modern day populations have focused on the genetic demographic history of Ethiopia and found patterns of linguistic stratification within Ethiopian populations, i.e., populations within the same language family are more similar to each other than to populations belonging to other language families. Pagani et al. 2012 and Lopez et al. 2021. Hellenthal et al. 2021. It is less clear if this pattern holds true to Northeast Africa as a whole, as Hohlfelder et al. 2017 found a stronger association between geography and genetics than between genetics and linguistic family. By studying modern day genetic variation, Molinaro et al. 2019 investigated the non-African part of Ethiopian populations and were able to conclude that there has been a Eurasian integration, likely coming from Levantine rather than Arabian populations. This event was estimated to have occurred around 3,000 years ago. That's long after obsidian spear tips, the spread of pastoralism, and EM78 emerging from EM35 in northern Egypt. Raising gene flow does coincide with actual historical events. Over the last 3,000 years, Egypt has been invaded by various groups, including the Hyksos, Cushites, Assyrians, Persians, Greeks, Romans, and Arabs. The Hyksos invaded around 1720 BCE, followed by the Cushites in the 8th century BCE, Assyrians in the 7th century BCE, Persians in the 6th century BCE, and the Greeks in 332 BCE. The Romans conquered Egypt in 31 BCE, and the Arabs conquered it in the 7th century CE. Egypt is and always was in Africa, and the genetic, archaeological, and cultural evidences show that it was inhabited by indigenous Africans who are still there today. Defining African solely as sub-Saharan is logically fallacious and commits the fallacies of biological essentialism and geographic determinism, just to name a few, Joshua.